Before we start in the next section, I have a question for you. Why do we need oxygen to live? It's not something we think about every day. We know we need oxygen, but do we know why? So I want you to pause the video, think about this. Don't cheat and look at the next slide, but think about it. Okay, so you might have said, we need oxygen to breathe. But we don't really need oxygen to breathe. We can breathe other things, but we die if we breathe those other gases, right? A large portion of air is nitrogen. So nitrogen's part of air. We can breathe nitrogen. But what does oxygen specifically do in here? You might think, well, it's in the red blood cells. And it goes to the cells. But what does it do in the cells? Well, this is one of the things we're going to talk about in this chapter. We need oxygen to survive. Us, particularly, there are microbes that don't need it, because oxygen is what we call the final electron acceptor in the generation of ATP by oxidative phosphorylation. So I talked briefly in the last section about the electron transport chain. Here, electrons are pulled through this, and they're pulled ultimately because they want to reach oxygen. That pushes hydrogen ions outside into the mitochondrial matrix, as it's called, and allows it to flow through ATP synthase, generating large amounts of ATP. But not every organism generates ATP this way. For some organisms, oxygen is in fact toxic. It's actually a very dangerous chemical to have around. It likes to react with things, and if you don't have ways of dealing with that, it can be very damaging to your cells. So we'll come back to this idea of oxygen versus no oxygen later in this chapter, but I just wanted to put that out there, that there are processes in here that you might not have thought about. So I'm going to start us with an overview here, and then we're going to dive into each of the pieces here. So we are ultimately looking at the breakdown of C6H12O6, which is glucose. In that, we will need to utilize some oxygen, as I just mentioned. So these are the reactants that are going into this set of reactions. On the output, we will get 6 CO2 coming out. You will notice that there's 6 carbons here and there's 6 carbons here. We're going to be chopping those carbons off of CO2 in these processes. Some water is going to be made, and ATP will be generated in the process. So, what are the 3 steps here? Well, for us, we're going to break it into glycolysis, which is our first piece. That's going to be like breaking glucose in half. Glycolysis, that means breaking sugar, so it breaks it in half. Then we have these 2 kind of related steps here. Taking pyruvate acid and putting it into the citric acid cycle. So we call this oxidation of pyruvate and the citric acid cycle here. Then the final piece is going to be this oxidative phosphorylation step that happens over here. So these are the kind of 3 big pieces. In my less beautiful diagram than that one, I've broken it down a little bit further because there are some alternative options to breaking down glucose. So glycolysis is always going to happen here. But what we do with that pyruvate could vary. There is kind of an escape out here to this thing called fermentation. This is an alternative path, but it doesn't actually produce any energy. But there are ways that microbes can live off of just these processes, doing glycolysis and then fermentation. We, on the other hand, we are multicellular. We need far too much energy to live off just this. We can do these processes, but we can't live off them. We take pyruvate and turn it into acetyl-CoA, sending it into a process called respiration. That involves the TCA cycle, which is the same as the citric acid cycle, or it's also called the Krebs cycle. Sorry, there's like a billion names for it. And that TCA cycle or citric acid cycle sends that to oxidative phosphorylation. So we're going to start by talking about glycolysis here. We're going to talk about the set of reactions that go into glycolysis. We won't memorize them or anything like that. But we're going to look at what goes in and what comes out, and a little bit about how some ATP is made in the process, and we will capture some high energy electrons in our NADH molecule. So this is the simplified formula going in here. We start with glucose, And through glycolysis, we're going to end up with 2 molecules that have 3 carbons. So I think my math checks out. 2 times 3 equals 6. So if we take a 6 carbon thing and break it in half, we get 2 3 carbon things. I think that makes sense. That's called pyruvate. We will generate 2 ATP in the process and 2 NADH. All right, now you're worried about these numbers here. Please, for this section, do not memorize how many numbers of ATP or NADH are being made. What we want to know is relatively, is it a little or a lot at each step? So this is a small amount of ATP being made here. So what is actually happening here? Glycolysis is a series of chemical reactions that lead to the production of this pyruvate and generation of NADH and ATP. Nearly every organism on Earth can do glycolysis. Okay, we all have the same enzymes for it. We think it's one of the earliest processes that likely evolved in life, and we haven't changed it because it's been working, right? No oxygen is needed for glycolysis to occur. And glycolysis takes place in the cytoplasm of both prokaryotes and eukaryotes. So this is not involving the mitochondria in any way. That's how ancient this process is. So what happens here? Well, we're going to have 2 phases. We're going to have kind of an investment phase and an output phase. So the first half of glycolysis, I'm going to show you the nasty reactions. Don't freak out. Okay, we start with glucose here. You can see all of these bonds in there. The first half of glycolysis involves putting in some energy. So this is investing energy. We have to spend money to make money, unfortunately. That's how the world works, right? There's no free lunch. So what happens? Well, glucose gets some phosphates added to it by an input of ATP. Okay, why am I showing you this? Well, I don't want you to memorize it. I don't even really need you to understand it too much, except for a couple of things. One, we're sticking phosphates on here from ATP, and we're producing ADP in the process. But you're like, wait, I thought we were making ATP. Again, we're investing some energy. We will have a payout at the end. Also, what I want you to take from this is down here, we're taking this 6 carbon molecule and chopping it in half. You can see this part goes there, that part goes there, okay? The molecule here is, this reaction is catalyzed by an enzyme. Each of these reactions is catalyzed by an enzyme, in fact. In the first one, glucose, we spend some energy from ATP, and an enzyme called hexose kinase changes it from a circular molecule to a linear molecule and tacks a phosphate on there. Then this one does some other changes, and then this one adds another phosphate on there, so on and so forth down the line. Okay, what to take from that? Well, each of the steps is catalyzed by a specific enzyme. That's what I really want you to get from it. All right, so this is our investment phase. We've added phosphates from 2 ATPs and chopped it in half. We end up with 2 3-carbon molecules. How about the second half? Well, in the second half, we are going to have an energy output. This is going to happen through substrate level phosphorylation. So we're going to start here and flow that way. So each of the 2 molecules that were produced, the 2 3-carbon molecules, would go through this process. I'm only showing one of them here. So this is times 2. So the first thing that happens is we actually extract some high energy electrons here. NAD plus grabs those electrons, becoming NADH. Something important to note here. If you don't have enough NAD plus, this really can slow down the rate that this whole thing happens. Oxygen or fermentation can help with a low supply of NAD plus. I'll come back to that a little bit in a later section. So we're gathering some high energy electrons. And in fact, we are adding another phosphate here. So now we have 2 phosphates. And we are going to then take those phosphates and add one of them to ADP, forming ATP. And then the other one we're going to add to another ADP, forming ATP. So now we're back at net, nothing has happened, right? But we multiply this by 2, and we've produced 4 ATP in total. That means 2 net ATP. So we invested 2, but we got 4 back, 2 net out of this. We've also got some NADH, storing high energy electrons, and we have these pyruvate molecules here. Now, like I said, this could be the end of it. We could go to fermentation and just deal with this, but there's stored energy in here. So we're going to keep extracting energy out of these pyruvate molecules. Okay, again, why am I showing you all of these steps here? Because each of those steps, it prevents that energy from being released all at once and keeps you from lighting on fire, right? When you eat a marshmallow, it doesn't explode because you break it down slowly, piece by piece by piece. All right, so we took glucose, we added some ATP, we ended up with 2 pyruvates, which are 3 carbon molecules. We produced 4 ATP, netting us 2, and we captured some high energy electrons. All right, let's take a look at this animation. Okay, this animation is more detailed than we need to know, but it's going to show you in a graphic way what is going on in there and give you a little insight to the complexity of it.
ATP is the key energy molecule for all biological organisms. One source of ATP is glycolysis, a series of reactions that break down sugars while producing ATP. Glycolysis is a series of 10 reactions that converts sugars, like glucose, into 3 carbon molecules called pyruvate. Glycolysis is an important pathway for cells because it produces ATP and other products such as NADH and pyruvate that can be used later to produce more ATP for a cell. This key process takes place in the cytosol of the cell. Let's take a look at an overview of the key steps of glycolysis and focus on the input and output molecules. The first 5 steps of glycolysis convert a 6-carbon sugar, glucose, into 2 3-carbon sugars, glyceraldehyde 3-phosphate. To do so, ATP is consumed in the first and third steps. Both of these chemical reactions are catalyzed by an enzyme known as a kinase, and both reactions are irreversible. The next 5 steps of glycolysis produce energy. 1 ATP is made per glyceraldehyde 3-phosphate in both reactions 7 and 10. This means a total of 4 ATPs are produced in glycolysis. Both of these steps are carried out by a kinase reaction. Note that step 7 is reversible, while step 10 is not. It is important to remember that the products in steps 6 through 10 are formed for each of the glyceraldehyde 3-phosphate molecules generated in steps 4 and 5. This means that a total of 2 NADH and 4 ATPs are made in glycolysis. Because 2 ATPs were spent in the early steps of the pathway, glycolysis results in a net of 2 ATPs. There is one more key product of glycolysis to consider, called NADH. NADH is formed in the 6th reaction by an enzyme called a dehydrogenase. NADH is an electron carrier with the potential to produce more ATPs through a process called oxidative phosphorylation. Details of that process are shown in a separate animation. As you can see, glycolysis is a process involving multiple enzymatic reactions that break down sugars. Its major contribution to biological organisms is the production of ATP, which is used to drive many other biochemical pathways.
All right, so like I said, more complicated than we need, but it's nice to see these pieces in there, right? Each of them catalyzed by an enzyme. There's lots of steps in there. There's lots of regulation in that. But ultimately, that's what's happening there, and lots of different organisms do that. So we took our glucose, and it went through glycolysis, and we broke it into pyruvate. We've made it this far, okay? We will continue on in the next section into these things, and at some point here we will take the detour and look at fermentation as well. So this process is occurring in pretty much all cells But in multicellular organisms, some do more of this than others. These later steps, they need mitochondria in animal cells. An interesting side note is mature red blood cells, they don't have mitochondria. So they get their ATP from glycolysis or from their surroundings, from other cells in the organism. So, but pretty much everything does glycolysis. All right, let's review before we finish. So what type of phosphorylation is occurring to make ATP in glycolysis? Go ahead and pause the video, think about that. All right, this would be substrate level phosphorylation because we're taking phosphates off of a molecule and adding them onto ADP to form ATP. So we started with glucose, a 6-carbon sugar that has energy stored in its bonds. The breakdown of glucose in humans is going to involve 3 main steps: glycolysis, oxidation of pyruvate and that citric acid cycle or TCA cycle or Krebs cycle, whatever you want to call it, they're all the same thing. And then the final bit, oxidative phosphorylation. Glycolysis is taking 1 molecule of glucose, breaking it in half to form 2 3-carbon molecules of pyruvate. This takes place in the cytoplasm, and it has 2 steps. We have the energy investment phase and then the payoff phase. The end result is 2 pyruvates, 2 ATP net, and 2 NADH coming out of that, storing high energy electrons. All right, in the next section, we will follow these pyruvate molecules.